In today's lecture, Ms. Nikita Gehoy, a final year PhD student in my proteomics laboratory at IIT Bombay, will talk to you about one of the applications of protein microarrays in cancer research. As you are aware that we can have different type of protein microarray platforms. Of course, if you need the purified proteins, that is much more laborious and big task. You can also have the genes printed on the chip, you can have the cDNA clones, you can have the tissue or the cell lysates printed, or you can have antibodies. You can have many ways of making arrays. However, if you have the purified protein based protein microarray, that can have lot more impact because it can have tremendous value for many applications. So, for this project we collaborated with a collaborator from Johns Hopkins, Dr. Heng Yu and his lab, and they have made a human proteome arrays, HuProt arrays, where all the possible proteins of humans so far, around 19,000 human proteins have been printed on the chip. And these proteins are printed in duplicate, and along with these proteins there are many spots which are control features. So, by using this kind of array platform, Nikita has tried to use for various type of brain tumor patients who are suffering from gliomas and meningiomas, how to screen these patients' serum sample on HuProt chip and try to identify the potential autoantibodies and biomarkers obtained from this experiment. So, in this lecture she is going to talk to you and provide you an overview of how an experiment using HuProt arrays can be performed, and also various details about performing such experiment, your experimental requirement, the protocol details, and finally about data analysis. So, the basic workflow of different type of protein microarrays are very similar as we have been discussing in different lectures. However, there are several small differences which you will notice when you are using an APA based platform versus you are using a purified protein platform, or you are going to use the reverse phase protein arrays based platform. Still, the number of proteins on these HuProt chips are much larger than the various other arrays platform which we talked, including the parasite protein based arrays for malaria research which we have discussed. Each slide which we are using here for the from the HuProt chips, those are used only for one patient sample. So, it means if you have an array on which there are thousands of protein printed, almost 40,000 spots are there. So, then only one patient sample can be probed on the whole arrays. Whereas, if you have a smaller number of genes printed on the chip, then you can have multiple wells and you can use many sample screening on the same platform. So, these are the small details which I am sure as we go along in the lectures, you will understand that how to do different type of experiment, also multiplexing capabilities. These things only will be clearer when we reach towards the end of the course, when we can discuss about comparison of different platforms. So, in this case when we are talking about the clinical sample usage on the entire chip, the sample handling becomes very critical, even the entire assay reproducibility become very crucial. So, these things are going to be discussed in much more detail. So, Nikita is going to demonstrate the entire experiment in the context of autoantibody signatures of brain tumors. So, let us have this lecture and try to understand the workflow involved using purified protein from HuProt chips and its applications in brain tumor research.
Hello all, today we will be talking about one of the applications of protein microarray in cancer research, which is to detect the presence of autoantibody in cancer patients. I am Nikita Gehoy and I am currently a senior PhD student working on detection of autoantibodies in cancer patients. So, let's start with basics of protein microarrays. So, if you can see in this slide, these are this is just a basic image of protein microarray slides, and these spots you see are actually the proteins that are printed on the slide. Majorly, protein microarray contains an array of a lot of proteins which are immobilized on a glass surface. And then, what are autoantibodies? Cancers are a cause of lot of mutations, because of which aberrant molecules are produced. Either they are produced in a higher level, or because of altered protein structures, or because the cell death mechanism, cells release a lot of differentially produced molecules which acts as antigen, and then body produces antibody against it. So this can be produced because of a large number of reasons, like neoepitopes, like the proteins are modified because of different splice variants and which body does not recognize as cell, and then antibodies are produced against it. Then sometimes there are proteins which should be present at certain concentration, but then they are produced at a larger concentration, and then body reacts against it and produces antibody against it. The post-translationally modified proteins, or sometimes it's the expression of proteins are supposed to be present in the in some different organ, but then they are expressed at other location of the body, which leads to production of antibody. So these antibodies are produced actually against your cell proteins, which now acts as foreign, and the immune surveillance detect them as something foreign and creates antibody against it. So in the study design of this experiment will be, we first take a patient cohort, we take sera or CSF from the patient, and then these are the chips which contains around 19,000 proteins, 19,000 protein spots and duplicates. So we incubate the slide with the sera, and then wherever the antibodies are produced against the proteins, this antibody will go and bind to the protein, and then we can detect the signal using Cy3 or Cy5 dyes. So let's talk about the experimental procedure. So the experimental procedure is very similar to that of Western blotting, where the slide contains a lot of protein molecules, and then there are a lot of spaces where the area is empty, and then first we do a blocking experiment where we use BSA and super block to block the extra spaces where protein is not available. Once that is done, we incubate the slide with patient sera and anti-GST antibody. Anti-GST antibody we put because each and every protein in this chip has a GST tag. So just for the quality control of the slide, every protein spot should bind with the anti-GST antibody, and we see a response against it. And against whichever protein the autoantibody is produced, the autoantibody from the serum will go and bind to it. So we use a cocktail of serum which contains the primary antibody from the human and anti-GST antibody cocktail to incubate it with the slide. After incubation for 2 hours, we wash the slide, and then we incubate it with the secondary antibody which contains Cy3 and Cy5 anti-antibodies, and then we scan it at dual channel. Now let's move on to the hands-on experiment of how autoantibodies are detected using protein microarray. So this is how the slide looks like, and this is just a normal glass slide which contains 19,000 protein spots. This is the area where the spots are printed in duplicates. This is the area where the barcode is present, and you cannot touch the slide where the spots are present. You have to majorly touch the slide here only. This is one of the precautions, otherwise fingerprints can result in erroneous results. So this is one of the slides which I am taking, and I am putting it in this box. This is another slide. This is the experiment where I am showing 2 slides that can be screened together for screening 2 different patients. So this is another slide. The first step is blocking of the slide. So for that we use BSA and superblock solution, and then this is the slide in the box. We will just pour the BSA and superblock solution onto the slide. Make sure that you put it on the barcode region, and then the slide is kept for incubation for 2 hours, so that all the extra spaces except for proteins are blocked properly. So this is done using a rocker, and we start it, and the rocker keeps on moving for 2 hours. The blocking is done. Speed is kept as such. The slide is covered properly with the blocking solution. So once this blocking step is completed, we take this blocking buffer out. We decant it, carefully decant the buffer in the discard. So make sure that the slide doesn't fall, and remove the blocking buffer properly. Once that is done, take the slide carefully without touching the protein spots. So for that, you can just touch the slide at the barcode region, and then we place the slide carefully into the cassette. So this is the cassette where we place the slide for washing. We will do the same for the another patient. So this is how the cassette looks like, and we keep the slide, keep the protein area facing each other. Now we take the cassette and place it into the buffer for washing. This buffer is 1x TBST and the pH is set at 7.4 so that the protein structure remains intact. Now we just start. So here you see there is a magnetic bead which is used for the circulation of the buffer. So we start the magnetic bead, and the circulation of the buffer makes sure that the slides are washed properly so that the extra blocking buffer which is present over the protein spots can get removed properly. This washing step is performed thrice for 10 minutes each. So once the first step of the washing is complete, for the 10 minutes, what we do is we flip the slide, we stop this magnetic bead, and then we flip the slide so that the washing is uniform from both the ends. Make sure that you touch the slide at the corners or at the barcode region, and then we flip the slide and place it again into the washing buffer. Same is repeated for the another slide. Touch the slide at the corners and place it back into the buffer. And now we start the washing again. So this is done thrice so that all the extra blocking reagent is removed and the proteins are now available for the primary antibody incubation. So once again, make sure that you close the beak before taking the cassette out, and then now we remove this cassette. And just to remove the extra buffer, what we do is we rinse it with water. So this cassette contains milky water. We take out the slide and then we rinse it in the water to remove the extra solution. The slide is then spun down to dry and to remove all the extra buffer. So we remove the water by placing the cassette on the tissue, and then we spin it at 900 rpm for 2 minutes. Once the slide is dried, we place the slide back into the boxes. And put the cocktail of primary antibody. So this cocktail contains serum and anti-GST antibody in BSA and TBST. So now this is done for two patients. So we will place the primary antibody in 2 different boxes. So for patient 2, we will place it onto the sample, onto the slide. Now again, this slide is incubated with this human sera and anti-GST antibody for 2 hours on the rocker. Once the incubation is performed for 2 hours, we decant the serum sample. Make sure that the slide is kept properly and we do not disturb it. Now we take the slide out. And put it back in the cassette. The same procedure is repeated for the second sample. Make sure that the magnetic bead is placed before you place the cassette into the TBST. Otherwise, the magnetic bead might hit the slide and we can get a blotch in the slide. So here you can see the magnetic bead is already kept with the TBST there. And then we start the washing again. So once this washing is complete, 3 times 10 minutes, we will again remove the cassette. rinse it with water to remove the extra buffer. And spin it for 2 minutes at 900 RPMs. The slide, once dried, is then again placed in the box for the secondary antibody incubation. So the secondary antibody mixture is a light sensitive reaction, and it contains Site 3 antibody, which is anti-rabbit, that is against anti-GST antibody, and anti-human Site 5 labeled anti-human antibody. So here we will put the solution onto the strip. So now we will do the same thing for the second patient sample. And then, since this is a light sensitive reaction, make sure that the slides are covered with aluminum foil or with some black box, so that the light doesn't interfere with the reaction. So then we will start the incubation for 2 hours in dark. After 2 hours, we will stop the reaction. We will do the same procedure, but then everything will be performed in dark. Once the slide is dried, we go ahead for the scanning of the slides. So for scanning, make sure that the scanner is on for at least 20 minutes so that the lasers are stabilized. Now, so this is one of the process slide which I am going to place inside the scanner. So in this scanner, we keep the slides in the inverted position, that is, the surface that contains all the proteins should be facing downwards. So this is the area where the slide is kept. This is the slide holding area. Place the slide opposite so that the protein spots are facing the lasers. Now we will start the software. We will start the scanning. So this is GenePix Pro software which comes along the scanner. So this is how the interface looks like. So now, because it was a dual channel experiment where we have used both the antibodies together, we will scan it at 635 for the Site 5 labels and at 532 for the Site 3 labels. But once we have set the parameters, now we can also adjust the PMT gain. So PMT gain tells you the saturation or the spot intensity of the slides. So here I will show you, I'll give you a demo how the PMT gain can change the contrast of the slide. I will demonstrate how the PMT gain can affect the slide intensities. So for now, if we can see, it is getting scanned at 350 PMT gain. Say suppose if I increase it to 650, you can see how the scanning has increased. Now if I reduce it to say 200, the intensity of these spots will be reduced. So for the PMT gain settings, we have to make sure that not all the spots are getting saturated. Also, we have to make a balance that we are not losing a lot of intense spots. So for that, there is an option called as Auto PMT. We have to fill in the details here. Once you press the apply button, it will scan at 3 different PMT settings and it will tell what is the best PMT settings for the slides to be scanned. So this is how the scanning parameters are defined. Once the scanning parameters are defined, we go to the scan area and then we scan the slide. So this is how the scan slides looks like. And here this is scanning of dual channel. This is how the one of the block look like. So you can see in the red channel how the spots are visible. Also in the green channel how the spots are looking. Now we will lay the grid onto the slide to know what is the location of which protein spot onto the slide. So this is how the grid looks like. Each grid is unique for each version of the slides. So now we take the grid and we try to place it to map where the proteins are. So this is how the grid is laid, and then we try to resize the grid for each and every block. So now you can see the difference between the two blocks. The software has tried to align all the spots with the protein location, and this is how the grid is laid. This is repeated for each and every block. So once this grid is laid, we then for each and every block, we then process the data, and then we get the data in form of .gpr file. Hope you have got a basic feel of how the microarray experiment is performed to detect the presence of autoantibody in the cancer patients. So this is one of the applications of protein microarray, which can be used to screen multiple number of proteins using a very small amount of patient sample, and to detect the abnormalities in the patients. Thank you.
Okay, so I hope you got a good understanding of how different type of microarray platforms can be used for different applications, although the underlying principle remains the same. At many of these steps, if you have done the basic biochemistry experiments of SDS page and followed by Western blots, they are very similar. The various steps are very similar, different washing steps are involved, and it depends on your application what you want to get out of these arrays. You have to provide the either purified protein for the protein interactions, or look at the serum samples for looking at the autoantibody signature, or you can think about any other analyte which you can use as a part of the primary antibody or the first step, and then it will be secondary antibody followed by detection of signal. So, depending on your application, you are looking at the PTMs, biomarkers, interactors. You can select the workflows, but remaining steps remain very similar in different type of platforms. So, this experiment you must have noticed that the slide handling becomes very tricky, especially during the washing steps, and particularly when you have multiple slides coming from different patient samples, they are all hybridized at the same time. So, handling multiple slides becomes crucial. However, it is really important to have multiple slides parallel processed, otherwise you are going to, if you process only 2 slides on one day and another 2 slides on tomorrow, then you are going to have much more variability as compared to if you screen 10 slides on the same day, then all the 10 patients get the similar kind of treatment, and your chances of getting higher reproducibility and better normalization can be achieved. So, we will continue this discussion about using the purified protein based arrays in the next lecture as well, and you will learn about how the microarray data could be analyzed, especially coming from such large data sets where many type of control spots become very crucial, and your considerations about how to ensure that what we are identifying is the right spot, it is not an artifact, it is not a false positive, and these considerations can only come when we talk in detail about data analysis aspect. So, we will talk to you about microarray experiment and same application and data analysis in next class. Thank you.